From Microbe TV, this is Immune, episode number 8, recorded on May 17th, 2018. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello and welcome back, listeners. It's beautiful in Ithaca. I think spring is finally here, or summer. We're almost June, but I got to eat outside today at the table after our new renovations here at the College of Veterinary Medicine, so it's really great.
So you know that every day trillions of viruses fall from the sky, right?
I do.
And so you probably ate some with your lunch.
I did. I'm sure I did.
Okay.
Hopefully none in my food.
Also joining us,
breathing them.
Also joining us from Worcester, Ohio, Steph Langel.
Hey now, things are good here as well. It's quite warm. I'm living that fifth year grad student life, just trying to get experiments done, get to graduating at the end of this year. So it's a combination of very busy, but exciting and scary, all wrapped in one.
That's life.
Do you have any advice?
Just do it.
Keep pushing.
Do what you want to do.
Enjoy being a graduate student while you're a graduate student.
Yes.
We're always anxious to move on to the next step, but being in graduate school really is a wonderful and amazing time, and just enjoy it while you're there. It's great.
Do you miss being at the bench?
A little bit.
Yeah.
Yeah. But it's also, I mean, each stage has its own excitement and rewards. I'm sure Vincent can attest to this. As you have more people working with you and they're making their discoveries, there's just more discoveries that come out of the lab, and you can see them from a bigger perspective, and you get to see the excitement that you enjoyed when you were at the bench too. And I still work at the bench occasionally as well.
I'm going to put that as like a positive affirmation on my bench, and I'll quote you. It'll say Dr. Cindy Leifer.
Absolutely.
I remember when I wrote my thesis, we had no computers.
Oh my gosh.
And I had to type it. And, you know, for figures, we would have images printed out on photo paper, which we would cut out and paste into the thesis.
I'm so happy it's 2018.
And I remember my girlfriend and I putting my thesis together. Now, my girlfriend is now my wife, so it turned out well for her and me. But we sat at the dining room table until 3 a.m. pasting the bloody pictures into the thesis to give out to the readers. Like I had to make what, 4 or 5 copies by hand. I had to make them all. I had to paste the pictures into each one because we couldn't just copy each one. So it took a long time. And after that, my dining room table was covered with glue residue, which I never could get rid of.
Quite the labor of love.
However, it was a dining room table that I found on the street in New York City, and it was in good shape, so we just took it and used it. But as Cindy said, every step is different. You know, I look back and I don't wish I were at a different time, and I think that's the way it should be. I think you have to embrace where you are at the time, and if you long for something in the past, something's not right.
Right.
Yeah. And you know, you lament about the glue that you had in pasting all your images together, but the students these days inevitably have a computer crash when they're in the last stages of their thesis. So to those who are out there listening, and to you, Steph, back up multiple places, email it to yourself, make sure you have multiple copies, because inevitably it happens right at the worst possible moment. You go to open your document or save it, and it says fatal error, and you're screwed. You need to go open a previous version.
Speaking of yesteryear, we got an email yesterday from Anthony. So yesterday was born on that day in 1845, so that's May 16th. Elie Mechnikov, the Russian zoologist who pioneered the study of cellular immunology. And he discovered your favorite cells.
Macrophages.
He got the Nobel Prize in 1908.
Yep.
So that's very cool. Thank you, Anthony.
Very nice.
All right, let's move to some science. And this month, May, it's Steph's turn. What do you have for us, Steph?
Yes, I'm actually going to be talking about one of my, well, I wouldn't say my favorite, but one of my favorite cell types because of its uniqueness and ability to work in the body, and the fact that we don't know a lot about it right now, is gamma delta T cells.
What is your favorite cell?
My favorite cell, well, it bounces back and forth. I'm actually becoming more interested in monocytes. I think Cindy is influencing me. But probably B cells, and then really gamma delta T cells are pretty high up there. So it bounces depending on what mood I'm in. Yeah. So I'm going to be talking about a paper in a little bit, but I wanted to give an introduction on gamma delta T cells and their role and function in the body and some of the unique abilities of it compared to its counterpart, which would be alpha beta T cells. And in the context of our body, I'm going to be specifically talking about adipose tissue. We did mention this. I think it was our first episode Cindy brought to us. Yeah, our first episode. The ability of certain cells to impact thermogenesis, and that's specifically important in brown fat. I'm sorry, that was my cell phone.
Oh, what a terrible ringtone.
Oh, it's so generic.
Just
so
kidding.
generic.
What does thermogenesis mean?
All right. So thermogenesis is the ability of the body to use heat, and specifically there's non-shivering and shivering thermogenesis, which I'll talk about in a little bit. But we're going to talk about gamma delta T cells in the context of fat and thermogenesis in this paper.
So I think we already have a lot of little lingo here. Are you gonna clarify exactly what alpha, beta, and gamma delta mean?
Yes, definitely. And I'll go on to talk about exactly what brown fat is compared to white fat. So I'll first just talk about the gamma delta T cells and the discovery. I mean, they first initially were looking at T cells as only having an alpha and a beta chain as a part of their T cell receptor, or TCR, as we have talked about it before. And a TCR, it's a heterodimer. It's made up of two glycoproteins that stick on the external surface of the cell. And of those, they have a constant region, which is at the bottom of this external portion, and they have a variable region, which is at the top. And it makes sense that the variable region is the part where the antigen is going to bind, because it's going to undergo some changes to be able to fit to whatever antigen it needs to bind to. And so before there was gamma delta T cells, there was only an alpha and a beta chain. And it was just this serendipitous discovery in 1987 when Chien and authors discovered the delta chain in addition to the gamma chain. And what they found, it was inside the alpha chain locus. There was this region that they found was rearranging early in the fetus. So in the fetal thymic development, they were seeing that there was this T cell receptor rearrangement happening before the alpha beta chain. And this new constant region, that was considered the delta chain, and then that paired with the gamma chain. So now we have these two T cell receptors, alpha beta and gamma delta. And it kind of then led into this really exciting phase where you had this unexpected discovery of the TCR receptor, but it was followed by years of uncertainty because this receptor was not binding in an MHC1 or 2 manner, which we have talked about before, major histocompatibility, complex, which is going to present an antigen to T and B cells. It did not respond to antigens in that way. And it's been a lull of trying to find, well, how do these gamma delta T cell receptors, how do they respond, and how does the T cell react?
But we know they should, because as you mentioned, they're rearranged like the alpha beta. So that means that they are generating different specificities of being able to recognize something, but we didn't know what it was. We didn't think it was protein presented by MHC, but we didn't know what it was.
Exactly. And the lull has kind of, I think in the later 2000s, there have been discoveries of certain things that the TCR of the gamma delta T cells do respond to, which I'll talk about in a little bit. And of course, they found it plays roles in pathogenesis of different bacteria and in cancer, which it seems that if you have a immune cell and you want some funding, if you can find it to be a role in place in cancer, you're probably going to be able to get more funding to discover what this T cell does. So just a little bit about, we've just discussed in the past that T cells, they are derived in bone marrow, and then they are going to migrate to the thymus. So T cells, T being thymus, where they are going to undergo further development. And again, like I mentioned, it's this early fetal development where you start to see the rearrangement of the gamma delta T cell receptor in specific phases of development. And so it appears then that there's this specific ontogeny, or this programming of the cell type, that may be important specifically to neonates. And it really demonstrates that we don't have a good grasp on neonatal immunity, and that for a long time we thought it was more that these neonatal immune systems were suppressed and that they were devoid in different ways, but it may just be that they're different. And gamma delta T cells really are a cell type we can look at to say they're actually quite active in the young neonate, and then as the animal ages, you see more alpha beta T cells. So in humans and mice, I think another reason why it wasn't paid attention to as much is it only represented like 1 to 5% at the maximum 16% in peripheral blood in adults. But when you really got down to looking at this microenvironment localization of these cells, there were huge representations of the cell in the skin, in the intestine, and in the reproductive tract. So they're, go ahead, Cindy.
Oh, I was just going to say, why do you think that that's so important? And the fact that we don't see them in the blood, but we do see them in all of these tissues where perhaps pathogens enter, right?
Right. So because alpha beta T cells are known as an adaptive cell that has to wait on the MHC presentation, these gamma delta T cells seem to respond in more of an innate way. They are faster responding than alpha beta T cells. They secrete in pro-inflammatory cytokines to help defend against pathogens, and also just to create a homeostasis. As we know, we don't want pieces of our commensals to leak into our blood system. And so having these cells at these barrier sites, these epithelial barrier sites, seem to be contributing to why they are there, to be able to prevent breakage of that lining for bacteria to enter your body. So when you start looking at these different subsets, then you have to wonder, okay, well, how are these gamma delta T cells behaving at these epithelial barrier sites? And I know for me, I first became interested in these cells because specifically when you look across different species of animals, they are more highly represented in animals like cattle and sheep and chickens. And I worked with cattle for a couple years when I was doing my master's, and they not only are more highly represented, but they're more diverse. They have more of those diversity gene segments, and so they can rearrange in more ways than humans and mice can. So it's a particularly interesting subset of T cells. And we know that while they don't respond to MHC, presented antigens, there have been recent developments of responding to lipid molecules, to phosphoantigens, small non-peptidic molecules that are basically metabolites produced from bacteria. Again, going back to the idea that they're responding to bacteria at these barrier sites. So there's a lot of questions.
They do or they don't need presentation by an MHC-like molecule.
They do not.
That's what I thought.
They do not. So they're able to respond directly to these different type of antigens. There's a whole field involved with finding what antigens can we give to culture systems of these specific cells to get them to respond. And a lot of times, if you give them cytokines like IL-23 and IL-1 beta, they can immediately respond by secreting something called IL-17, which we'll talk about. And that's a signifier of, we would expect this, for instance, for a cell to respond that quickly, we would expect a T cell to have been primed already by an antigen. That is just not the case with the cell type. So it's a very unique cell type. And the other thing is it does actually respond in certain disease settings. So it can respond to things like cytomegalovirus infection, to autoimmunity. So there's a wide field available to understand what these cells are doing in the body. So I think that would then lead us to talking a little bit about the paper we're going to be discussing in these cell types, roles in adipose tissue. I'm going to move on to adipose tissue unless Cindy or Vincent have any questions about gamma delta T cells in a general sense.
I'm all right.
Okay. Great. So when we think about adipose, there's defined two different types of adipose tissue. There's brown adipose tissue and white. So I think the abbreviations are just BAT and what? Or wet? I don't know. WAT?
Yeah.
Would it be what?
What?
And so
it's important that one is jiggly and one is not, right?
Yeah, the white adipose.
We think of fat as like somebody who's obese, and that's not what we're talking about. We're talking about brown fat, right?
No, that's not what we're talking about. Specifically, you can find brown fat more percentage-wise in newborns or in hibernating animals. Because they use that, basically we discussed it previously, but there's this protein called UCP1 or uncoupling protein 1. And what this protein does is during thermogenesis in brown fat is it's able to uncouple proteins that are moving along the mitochondrial gradient from the synthesis of ATP. And that allows it to be dissipated into heat. So essentially you're uncoupling oxidative phosphorylation from ATP synthesis, and that is what is producing the heat in thermogenesis. And so when thinking about that and fat in general, both brown and white, there are immune cells that are helping to control that thermogenesis. And I don't think this was particularly appreciated maybe 10 or 20 years ago. I know that when you think of fat, you don't necessarily think that there are immune cells there acting and behaving in certain ways. And so in adipose, you are mostly going to find innate-like immune cells. So this is like 80 to 90% of the immune cells you find in fat are going to behave in an innate way. So they're responding quickly. They don't need MHC presentation. And whether they're responding by a co-stimulatory molecule, depending on what that surface receptor is then what cytokine it's going to be secreting. So I'm going to name just a few of these innate cells. They're pretty neat to talk about. And they're these cellular subsets that are relatively new in discovery. I think, Cindy, I don't know if you would agree, but they're kind of these exciting, more rare subsets that behave differently than the traditional paradigm of immunology. So invariant natural killer T cells, we describe them as iNKT cells, and invariant meaning they have natural killer like surface molecules. So they don't have the traditional alpha beta TCR. I think it's the alpha chain that's more invariant than the beta chain in those cell types. And basically what it allows it to do, it can respond very rapidly to danger signals or DAMPs, these danger-associated molecular patterns, so that it can stimulate macrophages to come to help reconstruct the tissue during some type of insult or injury. So that's one. The other cell type that is involved in fat is the innate lymphoid cells, so ILC, particularly group 2. These ILCs, they're relatively new in discovery, but they are going to help to support secretion of type 2 cytokines. Type 2 cytokines, if you remember from last time, are things like IL-10, IL-5, IL-13, and these are going to help create this regulatory environment. So you don't want a pro-inflammatory environment in your fat, or essentially in your body in general, but in this type in your body, it's more dominated by these type 2 cytokine secreting cells. But there are other cell types that during obesity particularly, so they take obese mice and they see that there are other cell types like NK cells and another invariant T cell called mucosal-associated invariant T cells. They produce inflammatory cytokines. And why that's important is because if you're producing those, there's likely you're not going to be able to sense and metabolize insulin correctly. So there's a whole field about learning of these innate cell types and their contribution to diabetes, insulin sensitivity, and how we utilize fat. The one cell type, though, that has a lot of questions to be answered is gamma delta T cells. And so combining what we discussed, gamma delta T cells, and the role in adipose fat, adipose, is what we then are going to talk about in this next paper. And so just to prime a couple more things so that I don't introduce any terms that we didn't talk about. The T regulatory cells in fat are very dependent on a cytokine called IL-33. And they need that cytokine for maintenance and function and expansion, as well as the ability to have this thermogenesis, non-shivering thermogenesis, or using UCP.
So do you want to comment on the difference between shivering and non-shivering at this point?
We can. So, essentially, shivering thermogenesis is not going to use the UCP1, and it's going to use more. Correct me if I'm wrong.
Muscles, right?
Muscles, right?
Yeah.
And then not contributed by brown fat. And so non-shivering is brown fat, and then shivering is your muscles are actually, I don't know, would contracting be the right word?
Yeah, I think it's just using, like if you go run, 5 miles or whatever, you're going to warm up because your muscles warm up. So I think if you shiver, it's a way for your muscles to generate some heat.
Right. Right. So there's a lot of questions. There's what are gamma delta? Are they in fat? What are they doing? Are they contributing to T regulatory cell, which we need to be able to have this regulatory environment so that insulin sensitivity is on point and being metabolized correctly? And then IL-33, what is the role of that in this axis? And that would lead us to our paper.
Let me ask you a question, which
yes,
not really directly related, but I'm just curious. So if in say obese mice with lots of white adipose tissue, correct?
Mm-hmm.
Is there any quantitative difference in these cells in the adipose tissue as a consequence of having too much of it, or is just more of them?
You're asking if the frequency is different rather than just the number?
Yeah, is there some pathological condition associated with these immune cells that you've just described in an obese mouse compared to a normal mouse?
Sure. So I know in reading that they do have an increase in the cells that are producing those pro-inflammatory cytokines.
Mm.
Yes.
And so that would be things like NK cells. The other one was those invariant mucosal-associated invariant T cells. I think a lot of innate cells that are producing inflammatory cytokines, you wouldn't find many T regulatory cells.
Right. And definitely I do know that the macrophages are dysregulated when it's an obese mouse and contribute to the inflammation and the metabolic differences.
Because as you know, they're also more susceptible to certain viral infections, like influenza, which we know is also true in people. And I don't know if it has anything to do with the system within the adipose tissue or systemically,
Certainly, it's all connected. So when you have the obese mouse, the microbiome is different. The inflammation state is different. So the cytokine milieu is different, and the metabolic changes that come with that, the glucose tolerance, it's all different. So it's hard to say which exact aspect of that is contributing to that. That may be known, but I don't know. But there's definitely lots of different components of the whole physiology of the animal that are off when it's obese.
Right.
Okay.
Cool. So I will move on. We'll introduce the paper, and the title of this paper I'm going to be talking about is Gamma Delta T cells producing interleukin 17A regulate adipose regulatory T cell homeostasis and thermogenesis. And the authors are, the last name, it's Ayano Kohlgruber, and the last two co-contributing authors are Michael Brenner and Lydia Lynch, and they're from Brigham and Women's Hospital in Boston. And let's see, I think there's Trinity College in Dublin, Ireland. Am I missing? I think those are the two. Yes.
I think so.
Yep.
Now, the interesting thing here, a coincidence, is Lydia Lynch is very similar to Lydia Lunch, who is the lead singer in the rock group called Retrovirus.
Oh, really?
Seriously? Oh, that's funny.
That's cool.
Lydia Lunch. Yes. Okay. Sorry about that.
Is Lydia Lunch a scientist as well?
No.
Oh, okay.
But she has a name that's one letter different. That's why I'm.
Yeah, I wonder where she got that name of that band. She thought of it just because she was around science or not?
One year they were playing in New York City when I was teaching my virology course, and I told the class, if you go to the concert, I'll give you extra credit. And many of them did.
That's nice. Did they?
Yeah.
Did you go?
No, it was too late. It's like 2 a.m. or something. You know?
Oh my God.
But many of them went and they sent, pictures as proof. So I gave them extra credit. Just having a band called Retrovirus is very cool. I don't know if they even realize what it is, but the name is good.
There's a group called The Vaccines too. And
really?
Whenever I have my Google thing send me information about when there's updates on vaccines, it sends me the
Do anti-vaxxers listen to them?
I have no idea.
They probably get some type of hate mail by association.
Yeah, that's right.
Yeah.
Sorry, go ahead. I'm very sorry to interrupt.
No, it's fine. Interrupt. It's fine. So that's the paper title. Those are the authors. And so I introduced a couple of things we'll be talking about. But if I, Cindy and Vincent, bring up something I did not define to make it more user-friendly, let me know.
Give you trouble.
Yeah, give me trouble. So the first question the authors were asking, when they're looking at the gamma delta T cells and the lack of information in adipose tissue, they just simply said, what are the percentages, what are the numbers, and are they more abundant across, they looked at several different adipose sites, and they did in fact find that they are more prevalent in adipose than in peripheral sites. And they looked at the lung, liver, blood, spleen, and they found that they are higher in number and percentage. And what they wanted to know was, were these gamma delta T cells, were they circulating into fat and then circulating back out into the periphery where they're this transitory cell, they're migrating, or are they resident? And that just means that they are sticking around. They had seated in the fat and they are not moving, and they maybe can, these clones of gamma delta T cells are staying in a very specific microlocation.
And it's important because with immune cells, most of them are circulating around the body and they perform different functions depending upon where they are. But then other immune cells do stay put. And there are functional differences and consequences about the cells that stay put versus those that circulate. So it actually is an important and interesting question to ask that.
Definitely, because I think if you can expand tissue resident cells, and particularly I'm thinking about memory cells in the context of infection, but not only that, I mean just innate cells in general to prevent, to be that first line of defense. It's very interesting. And so what they did is they used different types of mice and found out that the majority of cells in there are tissue resident cells. So they're not recirculating like the cells of organs that I mentioned, lung, blood. I think liver had a higher contribution of these gamma delta T cells than other peripheral organs, but mostly the fat, high percentage of gamma deltas, and they are sticking around. They're not.
Some of the adipose sites are not so high, right?
Right. I mean,
and they overlap with liver, like sub-Q and perirenal. Right. They're really so. I wonder why that difference is there. What's going on?
Particularly epididymal adipose depot. It seems they tested a lot of different sites there, but they seem, you're right, to be higher than the others. And it could just be, again, what is the pre-programming of these cells once they exit the thymus and then are seeding into these fat sites, and how does that play a role as an animal develops over time? It's just all interesting questions.
And it's important to note that they're looking at white adipose tissue here.
Mm-hmm. Yeah.
Because they switched to brown later. It confused me a little bit as I was reading this paper because the title has the thermogenesis in it and they're talking about brown fat. Right. But the whole beginning of the paper, the first several figures are really talking about white adipose tissue. And I think that's really important to make.
Yes. I'm glad you mentioned that. You're right, because it is confusing. And then they're going to potentially play different roles. And right. We'll learn a little bit about the different phenotypes of these cells and how they behave. And so they're there. They establish, and so they wanted to know, what are the types, what are the populations, and in immunology we learn about phenotypes of cells by staining them with certain monoclonal antibodies, and they found, I'm not going to go through all the different surface receptors because it can get quite in the weeds, but one particular receptor that I find interesting is CD27. I think that's an important receptor to remember, because whether or not these gamma deltas have CD27 determines what cytokines they are secreting, and we'll talk about that a little bit later. So the next thing they did, okay, well, we know what the surface of the cell looks like. What does the inside of the cell looks like? And they wanted to look for a particular transcription factor called PZLF. PLZF, a little dyslexic there. And it's expressed in particularly innate cells. It gives them, if they're expressing this transcription factor, they have a more innate cell-like phenotype. So they respond quickly, they produce certain cytokines. And when they took PLZF deficient mice, they had a decrease in these adipose gamma delta T cells, but not in tissues or blood. So that was their first signal that potentially, yes, this is the transcription factor that would matter for these gamma delta T cells in adipose.
And I thought it was interesting to mention, so PLZF, for those who are interested, is promyelocytic leukemia zinc finger. But the mice that are deficient in this are the ZBTB16 deficient mice. And so that, I don't know why so many genes are called different from their protein counterparts. So the gene makes the mRNA, the mRNA makes the protein, and so the protein is PLZF, but the gene is EBTB16. So it can be really confusing when you read the literature that way, but that's what it is.
Exactly. And for example, IL-17 is easy. The gene is IL-17. So
right.
And
so
easy.
That's often the case, but not always the case. Right.
Right. And maybe now's a good time too. One of the ways they were able to look at PLZF is they used a knock-in mouse. And so they took that ZBTB16 gene and a knock-in mouse is basically it's a targeted approach. You take a desired gene and you insert it directly into a specific locus. So when you have a knockout mouse, you're somehow disrupting the gene, but a knock-in is you're putting specifically a gene into the locus, and they did a reporter gene GFP. And so essentially then wherever this gene was expressed, GFP was going to be fluorescing. And they were able to then look both at whole mount slides under the microscope, but then by flow cytometry as well, defining these populations by if they're expressing the GFP or not. So it's just another way of localizing where these cells are in the body.
And it's been a powerful technique to use in immunology because a lot of cytokine genes have been tagged with green fluorescent protein or yellow fluorescent protein or red fluorescent protein, so that we are then able to identify the cells that are making those cytokines without having to manipulate them. Yeah. And we can purify the ones that are making those cytokines. And in fact, it has revealed that often the distribution of cells that are producing a particular cytokine is broader than we initially anticipated. And so new populations of cells have been identified using those techniques. And so in this case, they're looking at cells that are making this particular transcription factor that is associated with innate-like cell types.
Exactly. So now that they've established a phenotype, okay, number one, they're there. Number two, they have a particular phenotype and they are more innate-like. They wanted to know, okay, well then do gamma delta T cells impose some type of regulation on T regulatory cell populations like the other innate cells of the adipose fat, which would be those INK T cells, and then the ILC2 group 2 cells. And so to do that, they used TCRD double knockout mouse. And so that would be knocking out the delta gene in the mouse, so they would not have gamma delta T cells because that chain of the TCR would be missing. And so once they did that,
can make normal alpha beta T cells.
Exactly.
They just can't make gamma deltas.
Exactly. And so when they did that, they looked at these mice at, I think that's 20 weeks, and they saw that they had lower amounts of T regulatory cells and lower amounts of things associated with T regulatory cells like IL-10, as I mentioned before, and receptors on their surface, particularly the IL-33 receptor. And so this is kind of a nice story, as you can see where I'm going. The questions were gamma delta T cells, T regulatory cells, and IL-33. What are they doing in fat? And so when you get rid of gamma delta T cells, they are going to decrease these IL-33 receptor expressing Tregs. And the other thing that was interesting, which was not true of the other cell types they looked at, was that these gamma delta T cells are increasing as the animal ages, along with the Tregs. And so that was always a question. They found that these other innate cells, they influence Tregs, but they're not increasing with age as the Tregs are. And so
they actually decrease.
They actually decrease. Right. And so you would assume, well, if they're decreasing, how could they be, performing this impact on these cells? So there's this concomitant increase in gamma delta T cells with Tregs in this temporal way, which is interesting. So further trying to define, I had mentioned before CD27 on the surface, and that helps to define gamma delta T cells by what they are secreting. So they wanted to do gene expression analysis because they wanted to try to define these gamma delta T cells into two different groups. And so by utilizing RNA sequencing and gene expression analysis, they characterize the transcriptional function of these cells. And they were able to divide them into two groups based on a couple things, CD27, and then their ability to express that PLZF transcription factor. When they further looked into those cells, as has been reported, if the cells were expressing that PLZF, they were also expressing a transcription factor known called ROR gamma. And that particular transcription factor is important for another cell type that expresses IL-17 called TH17.
TH17. Yep.
And I know we're throwing a lot at you, but you could go back and listen to this very slowly and maybe like write things down to create a map for yourself.
Hey, not when you're driving, though.
No,
but
not
the important thing I think we need to realize is that there are these not primitive T cells, but undifferentiated T cells. And then depending on what transcription factors they upregulate, they can differentiate into different functional types of T cells. And so those are those T helper. So we have T helper 1 cells, T helper 2 cells, T helper 17 cells. And so there are these transcription factors, which are key drivers of those processes. And so ROR gamma T is one that drives TH17 cells. And so it drives the cells that produce TH17. And so the fact that these cells also express that, the PLZF positive ones express the RR gamma T, suggests that they would be a source of the IL-17, right?
Exactly. And then they were able to show that in fact, that's right. If they have RR gamma, then they are these IL-17 producing cells. They also produce TNF, which is another cytokine that we're going to talk a little bit more about. We focus on IL-17, but it's good to know that IL-17 and TNF are kind of paired together. They work cooperatively to impact immune cells. So, but for now, you have PLZF7 positive secreting IL-17, and then you have PLZF negative, and they are acting more like these Th1 T helpers or NK cells, and they are secreting interferon gamma. So interferon gamma, IL-17, the majority of cells that are in the adipose are secreting IL-17. They are not the ones secreting interferon gamma. And so that's an important distinction. And they went into, I'm sure, a lot of trouble and experiments to define them because it matters in how they're going to behave in the tissue and how they're going to define them throughout the body. So, okay, now we have the phenotypes. And the next question is, well, how can these phenotypically specific cells behave around T regulatory cells? Do they regulate the populations inside the adipose? And so what they went ahead and did is they went and used IL-17 double knockout mice because they thought, okay, if it's the IL-17 cells in adipose, if we knock that down, what happens? And in fact, the Tregs decrease. Cells that were expressing that IL-33 receptor also decreased. And so they were able in this way to show that IL-17 is important for T regulatory cell in adipose. And one thing I could bring up here, this might be a good time. You know, when you knock down IL-17, you're not just knocking down gamma delta T cells that produce IL-17. You're also knocking down TH17 cells, which produce IL-17, hence their name,
right.
TH17. So the one thing about this paper, it doesn't fit into the story of gamma delta T cells, so maybe it wasn't in the scope. But if we're talking about the contributions of this cytokine, I think it's a little bit of a disservice to not bring up TH17. And maybe knocking out TH17 and controlling that, knocking out IL-17, I think you may see that cell type does play a role. And if you look it up, you can find TH17s in adipose as well. So just an interesting little side note.
Yeah.
Okay, great. So we know the cells are there. We get the phenotypes. We know how they behave. And yes, they are in fact regulating Tregs to help create this nice T type 2 cytokine environment. They go on to do a couple more experiments, which I'm not really going to talk about because it's just further diving into the specific of these gamma delta T cells. We define gamma delta T cells by the variable gene or the V gene arrangements. And in humans, there's 8 different segments and they're named based on that. And Cindy, correct me if I'm wrong, there's a lot of naming systems that were developed for these cells that are not, they don't cooperate with each other. And it's a bit confusing, I think, in the literature. So I'm probably not going to go into this aspect of it, but just know that they further define these based on these V gene segments.
Yeah, so the lingo gets just really bad sometimes.
Really bad. I can get a delta T cell
in immunology. We don't name things based on phenotypes. I always was jealous of the people who study Drosophila and flies. Like they have genes like deadpan because the fly just lays there and doesn't fly, or spatula because it looks like a noodle. I mean, immunology, you've got IL, blah blah blah, and V gamma, da da da, and it can be very daunting for those who are not in the field to try and understand the immunology literature. And right. So, picking up a paper like this would be incredibly difficult for those who are not in immunology and have learned all the immuno speak. So I hope that we are doing a reasonable job, and our listeners will let us know whether we're decoding some of this immuno speak. I know it gets a little hard because you can't leave out some of these transcription factors and these cytokines that are so critical for defining what these cell types are and what they do.
I'm hoping that with each little section, I'm going to bring up kind of what we reviewed. And at the end, I'll do a big executive summary and we can kind of together look at the things we learned and kind of talk about it more. Right. So then the next thing, if in fact IL-33 is important for T regulatory cell homeostasis and keeping them in the adipose and expanding their population, then how do the gamma delta T cells relate to the IL-33? And again, similarly, there is this temporal increase in gamma delta T cells, Tregs, and what do you know? IL-33 also increases in visceral adipose tissue in this age-dependent manner. So knock out the delta chain, you don't have gamma delta T cells, you see significantly less IL-33, but not in a tissue like spleen, which they're using as their kind of peripheral control. And so when you're utilizing these different knockout mice, you're knocking out the delta chain, you're knocking out IL-17, they're putting together this story that it is the IL-17 producing gamma delta T cells that are necessary for the increases in IL-33, because when you get rid of one, the other one goes down. And in immunology,
and we need IL-33 for the T regulatory cells.
Exactly. So that's the axis. If you have gamma deltas producing IL-17, the IL-17 is stimulating something to produce IL-33. And what exactly is producing that? What is that something? Because it can't just come out of nowhere. It has to come from something. So once the gamma delta starts secreting IL-17, it has to hit an IL-17 receptor on the surface of what they have looked at as stromal cells. So we know that in adipose, it's not just immune cells, it's not just, it's all different. It's connective tissue, adipocytes, but they looked at the stromal cells as the main source of IL-33. And they looked at that because they have the IL-17 receptor. So they thought, well, let's investigate that because it can respond to IL-17. They go through a lot of, I'm sure, agita to be able to define what stromal cells are actually responding. I won't go into that because it's different populations and they're named based on the expression of surface receptors. But they were able to define what those stromal cells were, and that when you give an animal IL-17, it's able to increase IL-33. I had talked about that TNF cytokine. If it's along with TNF, it actually helps to increase it more. But I don't know, this paper kind of just focuses on IL-17, but there are other cytokines that help to promote this IL-33 production by these particular stromal cells that they found. And it's interesting. I think, Cindy, we had talked about it before, but in this paper, you'll find a lot of increases in two different things, protein and mRNA. Mm-hmm. And they're not always working together. For instance, they, in some cases, found an increase in IL-33 protein, but not in mRNA and vice versa. So when you're reading these papers, it's important to know they don't always correlate, right? Because if you have an mRNA increase, you would expect there to be more protein, but if it's not translated and transcribed into protein, it's not always correlative. So just a little side note. At this point, I'll take any comments from the peanut gallery before I move on.
Are we going to find out why these changes occur with aging?
No.
What do you think?
I don't think they address that.
They don't address that. I mean, I think there's a lot of things that go into aging. Firstly, just the programming of ontogeny and different transcription factors that are expressed as the animal gets older, but also there's microbial colonization, which can direct different cells. I think that is a big question, and it's a big field right now where people are trying to determine why there's this age-dependent increase. And I think it's both those things. Developmental pre-programming, microbial colonization. Cindy, any others?
Well, definitely there are changes in the regulatory T cells over time as well.
Right.
So there's multiple types of regulatory T cells, and some increase over time and some decrease over time. And so the natural ones tend to decrease over time, and they're important for this homeostasis process. So as you age, you get more of specific regulatory T cells that regulate very specific antigen recognition versus more innate like regulatory T cells. So there's a shift in the balance there, and so does that play any role? I don't know. I mean, what they looked at in the earlier figure, and you mentioned this, was that there's an increase in the number of regulatory T cells over time in these mice. That's really the sort of earlier in life to midlife, but then as we get older, they will tend to decrease as well. So there's ebbing and flowing of these cells and how they're participating in the regulation. It's really complex. As you age, your T cell populations in general change. They shift from being more broad to more clonal.
Exactly. Yeah. So the next section really is looking at this thermogenesis part, which is in the title. So before I kind of go into that, is there anything we want to talk about?
No, not necessarily. I sort of was wondering where that came from. It's a little bit out of left field, other than the fact that they know that there's a connection between IL-33 and thermogenesis. But it seemed like almost two papers to me with the thermogen. So they're everything up to this point is looking at white adipose tissue and T regulatory cells and gamma delta T cells and white fat and the IL-17 and the IL-33, and then all of a sudden they shift to brown fat and thermogenesis and USP1, and it's a comprehensive paper.
It definitely is. And, it's kind of funny the way that the paper is organized. I don't know if that was something that was asked of them or if that was always, because it seems like the thermogenesis part is just this one big chunk at the end. Previous sections were kind of divided more up into these different subheadings. So, I don't know.
And it seems to be the thing that's the selling point of the paper, right?
Yeah. I know. I don't know. But I guess we could just hop into it. Sure. So that was the question then. If they know IL-33 is being impacted by the Tregs, or is being impacted by the gamma delta T cells. So they took TCRD deficient mice and they compared them, and they found that there's less IL-33 protein in brown adipose tissue. Although then when they compared that to the white adipose, the protein levels were not different in the white, but the mRNA levels were. So I wanted to preface that was because they're seeing this difference. And so they're making some assumptions. They're saying, okay, in brown adipose tissue, the protein levels are different. In white adipose tissue, only the mRNA. So maybe we could expect differences in thermoregulation. But for sure, in brown adipose, they saw a difference. And that corresponded with a lot of things we know about thermogenesis and regulation of body heat. So there were more lipids, less UCP uncoupling protein 1, lower expression of a bunch of thermogenic genes after cold challenge. So they take these animals and they put them at 4 degrees for a certain amount of time and challenge them and see then what genes are increased. And in white adipose tissue, again, more lipids, less lipases, which would break down those lipids, and lower expression of these thermogenic genes. So you can see that both the brown and the white is being impacted by not having gamma delta T cells. So,
and their cold challenge here is kind of on the short side, because the first paper, when we did the thermogenic paper in the first episode of Immune, we were talking about housing these mice from weaning until adulthood, and then analyzing what's different. And here they're challenging them for 4 hours. So this is like if we go skiing. So I wonder how we compare that, because brown fat really, as far as my understanding of this, is really an adaptive process in which the brown fat increases for over long periods of time when you're cold, and so it's generating this increased heat to maintain body temperature, whereas a cold challenge for a few hours is a little bit different. So I'm not sure exactly how to do a comparison there.
Right. And I mean, if you think about where we see an increase in brown fat is in animals that hibernate, and that is an extensively long cold challenge. So
correct.
Yeah, I think you're right. And I think that kind of plays out in their results here. So like if they were looking at the T cell receptor delta chain knockout mice, they did have lower body temp, lower energy expenditure, but they were able to recover. And so it actually wasn't until they knocked out IL-17 completely is when those animals could not regulate body temperature to the effect where they had to take those animals out of the cold challenge because they I guess were going to die. They were,
yeah.
Yes, they were moribund. And again, that goes back to, okay, gamma delta T cells could be contributing to thermogenesis, but it's the cytokine IL-17, which is not just produced by gamma delta T cells, produced by Th17 cells. And so I think this paper, they're very careful with their wording. So they, throughout the paper, talk about gamma delta T cells and IL-17. But then in the title, it says gamma delta T cells producing IL-17 regulate the thermogenesis. So I think I would need to see more proof. I definitely think it's a good paper. It tells a story, right? But longer cold challenge, let's look at the Th17 cells, I think could, for me, better define how thermogenesis is affected.
I think it's pretty impressive, their data showing that without IL-17, these mice die within 9 hours, like 100% of them. That's crazy. Lacking one cytokine, and they can't survive being at 4 degrees for more than 9 hours. 100% death. It's crazy.
It's crazy. And, when they tried to then show, I think they looked at also human cell lines, because of course this is all in mice and we want to make sure, I think similar things when they could stimulate with IL-17, you saw similar increases when you took it away. So you're right, though, with the mice, they did fare the worst. And I think further information about IL-17, in particular, that cytokine, could tell us a lot about how fat's regulated.
Yeah.
So that, that really wraps up the paper. I would say that there were kind of 3 take-home messages when we're thinking about these cells. So they found that it was that transcription factor PLZF positive IL-17 producing gamma delta T cells. They're able to interact with those adipose stromal cells to regulate IL-33 and T regulatory cells. So there is an axis interplay happening with those 4 things. Well, 3 things rather. I mentioned TNF. So TNF cytokine plus IL-17, if they're working together, that's the best ability that these cells are going to have to increase IL-33 to modulate by the stromal cells. So IL-17 is important, but you throw in TNF and it really does increase the ability. And then lastly, and there are, again, specific in the paper, gamma delta T cells and the cytokine IL-17 are critical regulators as separate entity. Well, together, I guess they only in the thermogenesis part looked at gamma delta T cell knockouts, not specifically IL-17 producing. And why this is important is because if we're looking at regulation of development, these cells are in greater proportion in neonates. you can find them increasing by huge amounts during disease and in cancer. And so this paper just contributes to further understanding how they act in our body, which could then give us solutions if we're trying to tweak the immune system to go in a certain way, whether it be in obesity, diabetes. Those are two huge areas. And I just like this paper because I like gamma delta T cells. They seem like a type of cell where there's so much to learn. I mean, alpha beta T cells, talk about those all the time, but gamma delta T cells, there's so much to learn. So
IL-33 is the cytokine that's having some effect on thermogenesis, right? And what it is, we don't know.
Exactly. That's produced by the stromal cells by responding to IL-17, right?
Now, wouldn't you have liked them to inject some IL-33 into these mice and see if they could survive the cold?
Yeah. I think
that would be a great experiment.
I think that the fact that the thermogenesis is in the title would have warranted much more, I think, expansion, because it was just that one section. Who knows the story behind that? But yeah, that would have been interesting for sure.
Because then you would say that is what is causing this phenotype, even though, it's gone. 33 is very low. Would be nice to show this is actually what we need. And also to look at some metabolic activity, right?
Mm-hmm. Yeah.
In these animals. So is it known what IL-33 actually does to allow thermogenesis?
I don't think it's known.
I'm trying to look up if I can find something quickly about whether IL-33 is inducing the UCP1 and the other genes. I'm not sure.
Now, is the reduction in IL-33, this is systemic, I would assume, right? It's not just in adipose tissues.
Yes.
So, yes, right. But yeah, they focus right on the adipose, but correct, right. So that's the paper.
I think I just found something that says IL-33 regulates UCP1 expression as well. So it may be doing it directly.
But you're right, Vincent. If they injected and then saw an increase in UCP1 after a cold challenge, that would be...
Yeah, that would be interesting. Right. Yep.
Cool.
Now, why should the person on the street care about this?
Okay, well, overall, gamma delta T cells, they are important because if we're going to fight off certain infections, we need to know, can vaccination influence their increase? Can, are they important in memory? It doesn't seem, they're more innate cells, but it's good to know, particularly though in cancer. They are a targeted cell type to be able to kill cancer cells. They increase during cancer. And right now it's a really hot area to be able to engineer gamma delta T cells as just another cell, because we talk about T cell usage in cancer treatment, but gamma deltas, they react faster. So if you have a faster reacting cell that does not require MHC, it requires MHC-like proteins and co-stimulatory molecules, it's going to work potentially faster. So I think the hot area being cancer is one. And then secondly, diabetes and obesity. Those two areas.
So you don't think they should care about the thermoregulatory aspect of this, right?
I mean, if I'm being honest, I don't know. It is interesting, but I think what they're gearing that towards is how we are, not so much how are we keeping cold within 4 hours being put in going skiing, but I think it's more how is fat being utilized? Because that's the interesting. I mean, how are we regulating fat when and we have a huge population of people who are overweight. We have insulin sensitivity, diabetes. So I think that is more important than just brown.
Got it.
Yep.
Okay.
And wasn't there a big push to how can we expand our brown fat?
Yeah.
Because marathon runners have a lot of more brown fat.
Is that right?
Well, they said that they were in the paper. It did say that highly active adults have higher percentage of brown fat.
Yes.
Who run a lot.
How about those of us who think a lot? No.
We'd have to talk to a neurologist. See, there's brown fat of the brain.
We have a lot of neuronal connections, I guess.
Yeah.
Okay.
Cool.
That's cool. Thank you.
You're welcome.
Want to read a few emails?
Sure.
Yes.
The first one is to you, Stephanie.
Okay, I can answer. Okay, this is Deirdre. She writes, "Hello, Stephanie. I hope this message doesn't bother you. But after listening to the podcast regarding the lymphocyte trafficking paper, and I think that would have been episode 2, I had a few questions. Am I right in thinking that the paper loosely outlines the treatments and immune responses which occur at Z5? And just for reference, Z5 was just 5 hours after the day.
Onset of light, right?
Yes.
After onset of light.
Thank you. Onset of light. So immune responses that occur at Z5 are more fruitful in their outcome because there are more antigen-presenting cells and lymphocytes in the blood system to not only recognize the antigens, but to also carry out their effector role when dealing with a pathogen." And so, yes, she is correct in that paper. They did demonstrate that lymphocytes, and they looked at dendritic cells as their APC. In the periphery, during the day, there are more, and then there are more lymphocytes in the lymph node at night. So she's talking about how can this impact during a viral infection. And I remember from that paper, it was just at the end, they briefly did a couple things. They challenged mice with influenza, and they did see higher functions during the day, specifically CD8 T cells. But in context, I think with circadian rhythms in mice, they have different circadian rhythms than humans. And there's a paper that just came out for pigs too, looking at these diurnal the changes in lymphocytes. So she's correct. It just, I think, depends on a little more information on how the species affects it.
Next one is from Kang, who is a colleague of mine here in my department.
Cool.
Huh.
She writes, "Happy New Year." So this has been in the queue for a while. The best thing over this winter break for me is to go back and listen to TWIV, which then led to your new podcast, Immune. I listened to all 3 episodes and enjoyed them so much. I should say Kang is an immunologist. She trained with Ralph Steadman and then Michelle Nussenzweig.
Oh, yes.
So she is a dendritic cell person. A big thank you to 3 of you for spending time to do such a great job. I love the way the 3 of you work together, particularly having Stephanie, an aspiring graduate student with such fresh and inquisitive mind. You make immunology sound fun and not intimidating. You're not an aspiring graduate student. You are a graduate student.
Yeah, I guess aspiring scientist.
Yes. Here's a comment on lymphocytes after dark. The oscillation, is that number 1?
2.
The oscillation of lymphocytes in and out of the lymph nodes is really nicely related to the influence of circadian rhythm on hematopoietic stem cells and their niche in the bone marrow. Check out work from Paul Frenette's group. Here's a new read suggestion. A new paper contributes to categorizing adipose tissue as a new immune organ.
Yeah, we love adipose tissue now. Yep.
And this is from Yasmin Belkade. White adipose tissue is a reservoir for memory T cells and promotes protective memory responses to infection. And have any of you seen that paper?
Yeah,
I had not read it, but I saw it.
Yep, similar.
And then it was moving to listen to female graduate students' conversation with a mother regarding family life and science. Here's a link to a lovely article in Nature Immunology by Akiko Iwasaki, an outstanding scientist and loving mother, balancing family life with a science career. Looking forward to new episodes. Warmly, Kang.
So if I am correct, I think Yasmin was just recently, or last year, made it into the National Academy of Science. Is that right?
I think you're, so. Yeah, this year. Yeah.
And Akiko just won.
This year.
As well.
She did.
This year. Yeah.
You bet.
Both fantastic immunologists.
Absolutely.
Wonderful women. Great role models.
Yes. And you as well. And as a mother in science, I mean, do you have anything to comment? I get a lot of questions of freaked out graduate students, like females. How am I going to do it all? And you seem to be doing it.
Yeah, it's not easy, but it isn't easy for anyone. But if you have a passion, we're problem solvers, and women particularly are excellent problem solvers and figuring out how to keep all the balls in the air at the same time. And they say it takes a village, but it just takes a group of people that help each other, and you make friends and keep them close and use them. I think, if I had to say one thing, I think women particularly have a need to do it all They feel like they shouldn't ask for help. They have something to prove. And when push comes to shove, we're all feeling the same thing. And if we just asked each other, we'd find out that, it's okay to ask, and it's okay to get help. And people are more than willing to help you so that they can get help later. And we don't use that enough.
Agreed.
All right, let's take one more. Cindy, this one from Anne.
Anne, okay. Hello, Immune team. I'm a crossover listener. It seems that's quite common there from the Twix series of podcasts, and have been enjoying this podcast very much. This episode describing CAR T cells was informative, and while I didn't understand everything, Dr. Leifer did a good job of explaining the complexities. Thank you, because it isn't always easy. So I appreciate that feedback. Several years back, I attempted an online immunology course and was so put off by how the terminology was presented. It seemed like an information dump with so much memorization needed that I abandoned the course. I understand that immunology is complex and that the designations of cell types is historical, general names based on less well-defined or more general functionality. But the lack of specificity or clarity in the names makes learning about them challenging. But I've wanted to develop a broader understanding of the subject, so I'm hoping this immune podcast will help. And so far it has, ! My very rudimentary knowledge of some of the terms used in this podcast was reawakened, and Dr. Leifer's explanations helped develop a clearer picture of the processes she was describing. No doubt the repetition of these explanations will help the listeners like me become more conversant in the basic terminology and processes learning. So I want to stop at that point, because I want to say we're often worried about repeating ourselves because we don't want to bore people. But I think we need to keep that point in mind, that some of our listeners don't know all of these intricacies and these nomenclature idiosyncrasies. And so we should remind them frequently. So we're going to have to keep ourselves on our toes for that. Okay, so back to this. I was intrigued to learn about the use of viruses to help bring some cancers under control, and spent time reading about poliovirus used to help cure people of their cancers. And they provide one example, which I have to admit I didn't have an opportunity to look at. I wasn't aware polio was being used. I know that there's some oncolytic viruses, which means viruses that can lyse cancer cells. And so I know that has been an area of study of interest in virology as well. So on the podcast, it was also mentioned that this CAR T immunotherapy could be used to target latent viruses, but I didn't find as much information about this topic. At some point, I hope you can discuss this. There are so many latent viruses, the HSV family of viruses that lead to recurrent infections, and shingles, hepatitis B, and HIV, for example, that being able to reduce their impact seems a worthwhile endeavor. Thanks again for all the podcasting learning opportunities. It's much appreciated. Looking forward to more podcasts. Regards, Anne. So I don't know if I know all the details about how these CAR T cells can be used to target latent viruses, but if the latent viruses are making some latent transcripts, so they're making some proteins while they're latent, those proteins could be targets of recognition by these CAR T cells that could then lyse virally infected cells. Now, this would work if not all the cells are latently infected, but a certain population of cells are latently affected, and that those that are latently affected are not critical for life. So I think that those are two issues, but potentially one could target HIV reservoirs, for example, if the HIV is making a glycoprotein or something while it's latent. And I'm not sure if it really does that.
Yeah, that's the problem. It doesn't, right? It's completely silent in these,
right.
Especially in the hematopoietic progenitors, they're silent. Yeah. So, yeah, you can't, unless you turn them on in some way. And a lot of people are trying to figure out how to do that.
Yeah.
Now, if you can get the virus into replicating, it will then kill the cell anyway. So you don't need CAR T-cell therapy for that. So, and of course, herpes simplex viruses are silent in peripheral neurons, so that won't work.
Right.
So
there's some limitations as to what one could do with that. So I may have mentioned that because it was in a review article that I read, but practically I'm not exactly sure how that would work.
So this polio therapy is interesting. This is a very well-known 60 Minutes episode where they had done a clinical trial on a couple of patients with glioblastomas, which are very serious, and they have a lot of patient reaction, and it's a very moving episode. This virus is very interesting. It's normally a neuronotropic virus, and this particular strain was modified. They took poliovirus and substituted a small part of the viral genome with that of rhinovirus, and this changes the tropism to these tumor cells. Just happened. It's not like you could plan this, but it was just an observation. And so this has to be injected intratumorally, right into the tumor, and the virus replicates. And now I understand that, frankly, I think some other kind of therapy is going to be better than this, because first of all, we're about to eradicate polio, so I don't know what we're going to do with this, because you can't have polio anymore after a certain point. So are they going to make exceptions for people with these tumors? I don't know, but nobody talks about that yet. This work continues to be supported, and it doesn't seem to make a lot of sense to me. But there are many other oncolytic viruses that are amazing out there. And we've done a couple of podcasts on TWIV, episodes on TWIV about those, which Anne has probably stumbled across.
Yeah.
All right, let's do some picks of the week. Steph, what are you thinking about this week?
Yeah, so I had seen this article about polycystic ovary syndrome, and I just am interested in pregnancy in general, as a lot of people know, and this it's a really it's the most common cause of female infertility. But what was striking is we don't know a lot about it. We don't know what causes it. And so this is just an article of talking about some recent work describing why they think this is happening. They found this hormone, anti-Mullerian hormone, is increased in pregnant women with this polycystic ovary syndrome. And they found that their offspring, their female offspring, showed signs of polycystic ovary syndrome. So there's like transgenerational impact of women who have this high levels of anti-Mullerian hormone. So that's kind of going to be a little bit of my reading this weekend. And if a lot of women struggle with that, I think it's neat to find that maybe we're finding why, and can we prevent it?
You know, do you like this New Scientist in general?
I don't think I've seen a lot of kind of like grabby headlines. You know what I mean?
Yep. Yeah. And also the ads are terrible. You go you go to the bottom, and this happens on a lot of websites, just non-scientific ads, right?
Yeah.
And they pop up, and then you have to try and find the X to close them.
I mean,
the article.
I know you have to make money, but if you're a science site, please try and do something more sciencey. But the article is interesting. Thank you.
Yeah, sure.
Cindy, what do you have?
So I had the pleasure of seeing Jim Cebulski speak here at Cornell for our department about a week and a half ago. And he was a graduate student with somebody named Manu Prakash. And together they invented a $1 microscope that is made of paper. And I got to play with one, and it works. Well. It's amazing. So I encourage people to visit this foldscope.com. So it's foldscope.com. And you can also watch Manu's talk, a TED Talk, where he talks about how he made this. But the idea came from being able to use it, for example, to use diagnostics, or their big thing is for citizen science, or for getting kids interested in science. So how can we get a microscope in every kid's hand type thing? And how can we get in countries where they don't have the money to be able to purchase $20,000 microscopes to be able to teach kids things, how can we get this into their hands? And so they were able to design this microscope that you can make. It's like origami. They call it origami microscope. And so you get this kit, and you put it together, and it's got a 20-cent lens which, I mean, there's limitations to this. Don't get me wrong. You're not going to get the images that we see in the papers that we review, but you can put this thing together, and it's waterproof because it's made of synthetic paper, and it costs basically $1 a microscope to make. And so it just opens up the opportunities for people children in Africa to experience science where they wouldn't have had that before. And so I took my phone, and you can like put it on video, and you can take images of things, and you can put pond water in this special slide that they have, and you can actually take videos of it. It's really pretty amazing. And they're creative because they're now working on some sort of centrifuge based on a kid's toy. Have you ever seen those kids' toys where you pull the string and the thing spins faster and faster and faster?
Yeah.
So they're making a centrifuge based on that. So now in the field, you'd be able to use this thing. It's your own muscle work, but you spin down some blood so they can separate blood, and then you can put it on this slide and then look at it in this $1. So for $1.20, a 20-cent centrifuge and $1 microscope, you can do this analysis in the field. And it's not going to replace what we have now, but it opens up the opportunity for kids across the world who might not be able to experience science to be involved, and then to potentially have some new hardware for diagnostics in the field. And one of the faculty members in my department, Ted Clark, is using it in teaching high school students and things where they're pulling pond water out, and they're looking at what's in there, to look at the health of ponds and things. So it's just, I thought it was totally fascinating. And if you have an opportunity to go visit the website, there's pretty cool videos on there as well.
Yeah, it's great. I think
very cool.
Give a kid a microscope, it's just...
Yeah, and you just open up their mind.
Yeah, and you don't have to tell them, be careful, this, that. Go ahead. If you want to break it, that's fine, right? That's the key, because then they can do what they want, and they'll discover things. It's
yes,
cool.
Exactly.
All right, my pick is my virology course this year. It just finished, and all 24 lectures are online at YouTube. And so those of you who listen to Just Immune and you want to learn some virology, go over there and check it out. The entire course, and you can watch it for free, courtesy of me and Columbia University.
Oh, that's awesome.
That is great.
That's awesome.
Steph, we have a listener pick. I think you should do it.
Yes. So one of the listeners is my husband, who's an optometrist. And, I don't know if this counts since technically I told him we were doing the gamma delta T cell thing, but he did send me a paper that's been kind of passed around in his field. And, one of the questions of the eye in eye immunology is, are there commensals? Because the thought is that having any type of bacteria in your eye being an immune privileged site, cells are not infiltrating and acting in the same way as other areas of the body, that there might not be commensals. Well, that may not be true, as this paper showed that there was a commensal that was able to be isolated. And guess what? It drives the proliferation of IL-17 producing gamma delta T cells. So another way that these cells are involved in the eye. So I thought that was interesting, and he did too. So pass that along.
So he listens to Immune?
He does. He'll listen
choice?
To it. Yeah. And it's embarrassing. I mean, I'm like, oh my gosh. He's like, listen with me. I'm like, oh, my voice. And, I do. We listen together though.
I see people walking down the hallways who have earbuds in. And the one time I said hi, and the guy popped his earbud out, and he goes, oh. And I said, what? He goes, well, I'm listening to you right now.
It's funny. Good.
Yeah.
The more, the better.
Exactly.
And that'll do it for Immune number 8. You can find it at microbe.tv/immune. That's where we put all the show notes that we refer to. Of course, if you listen, you're doing it on a mobile device, most likely, and there you use a podcast listener, and whichever one is your favorite, please just search for Immune and subscribe to us so you get every episode. They're all free, so why not get it? And if you have questions or comments, please send them to immune@microbe TV. And finally, if you really love what we do and want to help us out, consider supporting us financially. Like $1 a month would really help. Just go to microbe.tv/contribute. Cindy Leifer is at Cornell University. She's on Twitter as Cindy Leifer. Thanks, Cindy.
Thank you. Stay curious.
Stay curious. Steph Langel is at Ohio State University. Stephanie Langel on Twitter. Thanks, Steph.
Thank you. This is great.
And I'm Vincent Racaniello. You can find me at virology.ws, Prof VRR on Twitter. The music on Immune is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.